This is going to be a review of the AICE Marine Science October-November 2025 Paper 1 Time Zone 2. And this is for AICE Marine Science, and as it is a Paper 1, this is going to be covering the AS level, and this is the first paper that's pretty much comprehensive of the syllabus, or referred to as the theory paper. On the front, you'll put your name and then the center number and then your 4-digit candidate number. For this exam, we're going to use pen, black or dark blue. We are not going to use a gel pen or erasable pen. We're going to use pencil for any diagrams or graphs that we need to do. There will be no graphs on this paper, but if there's anything that you need to label or something that you need to manipulate or put a mark on that you cannot correct if you make a mistake, then we want to do pencil. Okay, you can use a calculator, and you should. You can also highlight or use a different color item to identify your brackets. That's where your marks are. Not every question will have a dotted line with it, so not every question is obvious that you need to answer it, but it is obvious if you look for the marks. This test, as well as paper 2, both of them are an hour and 45 minutes, and they are 75 marks. Okay. This first section is a nice, brief, comprehensive cover of 1.1. So they show us some diagrams, and then we need to find what is familiar about them, or at least associate them with the questions here on area A. As we go through them, even though just for review purpose, even though it's not part of the actual question for A, I'm going to go through each one, and from the syllabus, we need to know their name, we need to know their formula. We also need to know if they're ionic or covalent. One second. Hey, Olive. Yeah. Will you turn that down a lot? Mm-hmm. Thank you. Okay, sorry. I don't edit these, so. Okay. This first one is sodium chloride. Makes the lattice structure. It is NaCl, and it's ionic. That means it gives and or takes electrons in order to have full outer shell. And by giving or taking an electron, this is going to have a charge, a positive or negative charge, causing it to turn into an ion. Substance B is water, H2O. This is covalent. They share electrons. Here's your H, O. These are your little NAs. These are your CLs. Over here is CO2, carbon dioxide. And this is also covalent. They share electrons. D is calcium carbonate. CaCO3, and this is ionic. And then here is our glucose. Makes a ring shape, C6H12O6. And this is also covalent. We have three salts total for the syllabus, and one of them is here for A and also a D. The other salt that is not featured is magnesium sulfate, and that is an ionic bond. And then on here we have 3 of our covalent bonds. The other covalents that are missing is oxygen and sulfur dioxide, mainly from hydrothermal vents. That's where we're going to see that the most. All right, state the letters from 1.1. Identify the substance or substances that are products of respiration. We needed to go into this with the photosynthesis formula and obviously the respiration formula. But if you know one, then you can figure out the other. And I'm going to do the photosynthesis formula. So we have some light energy and CO2, water. Here's our light. And it's going to yield oxygen, C6H12O6. And then to nail down respiration, just flip your arrow. So in respiration, we eat the glucose, we breathe in the oxygen, we don't need light for it, and we get out ATP, we breathe out water and CO2. So if we know photosynthesis, we just flip our arrow and that's going to be the respiration. And if we're flipping that arrow, products of respiration, then is CO2 and H2O. And so CO2 is letter C and H2O is B. You could also put B and C, doesn't matter if that's the order. Your salts is going to be A, sodium chloride, and D, calcium carbonate. Salts are ionic bonds. You could also go D and A. Can form hydrogen bonds. Water is the one that we need to know. Forms hydrogen bonds. Our hydrogen atom compared to the oxygen atom, it does not have as many electrons around it, so we say it's partially positive. Oxygen's partially negative. And it'll form a hydrogen bond with another water molecule. I love that it does that. Phenomenal. All right. OH and H. And so our hydrogen bond is going to be right here. For example, hydrogen bond forms between a partially positive hydrogen atom and a partially negative oxygen atom of another water molecule. So partially positive hydrogen, partially negative oxygen atom of another water molecule. And those are weak bonds that are easily broken by kinetic energy, including heat. All right, this is going to be B. Has a formula calcium carbonate, CaCO3, that's D. And is a product of photosynthesis, so the result of photosynthesis, E. Describe the evidence from figure 1.1 that shows that the substance has covalent bonds, so sharing electrons. And the thing we need to look for, this is in section 1.1 of the syllabus, they talk about this overlapping of orbitals. So we need to see the shell overlap right here. That is showing that they're sharing electrons. The way that they did this is the electrons that are coming from oxygen are just these black dots. Oxygen is atomic number 8. And so it has 2 electrons in its first shell, making that first shell full. And then 1, 2, 3, 4, 5, 6, and then a total of 8 with those first 2 in that first shell. It's in group 6, so there's 6 valence electrons in the outer shell. It wants 2 more electrons to have a full outer shell. Hydrogen is the one that has the X. Hydrogen is atomic number 1, and it only has 1 electron. It only has that first shell, and so to have a full shell, that first shell can only have 2 total, so hydrogen would only require 1 more electron. And we share a hydrogen, each hydrogen shares an electron with oxygen. We need 2 hydrogens total. 2 hydrogens total to make the electron have a full outer shell. They both want full outer shells though. All right. E negative electron. Can you do that on a test? Yes, you can. That is like science. It's not something that I made up. But if you're not sure, if you ask yourself, am I allowed to do that? When in doubt, just err on the side of caution and don't. Spell out electrons. Electrons are shared between atoms. I also would maybe mention that the orbitals or the shells, same thing, overlap where the electrons are shared. That's sloppy. Okay. And now's a good time to mention, if you're thinking about it, can you put BTWN? No, don't write between. Not like that. The only things that you can do shorthand that you can do feel confident about are going to be electrons, protons, neutrons. Symbols for measurements and units. Okay, and that had a total of 6. We had a 5 for this first part and then a 1 down here. We're moving on. A dichotomous key. We see some different drawings of species of crustaceans. I already copied this image and put it down here so we could be able to see it all at once. All right, dichotomous key can be used to identify different species. They already identified species F for us. Complete figure 2.2 with the correct letter for each species. So just as a quick review, I am going to redo species F so we can see why they chose that. For dichotomous keys, we always start at the first option and then the number 1 and the first option within that. So the very top of the key. And it says there's more than 10 legs visible. I'm looking here. 1, 2, 3, 4, 5. And that's just not 5 right there. We know that there's another 5 matching on the other side. So there's 10 little swimmerettes right there. And then we have 1, 2, 3, 4. There's one that's overlapping I'm not seeing. 5, 6, 7. I know there's some that I'm not seeing on there. Period, we have more than 10. More than 10, it says go to 2. So then I go to number 2. One pair of antenna is longer than the length of its body, or the antenna is shorter than the length of its body. And we see here it's a much longer than the length of its body. So yes, one pair of antenna are longer than the length of its body, and that is going to be its scientific name, Penaeus monodon. We can identify that it's a species name because it's written in italics, and it always goes genus followed by species. You do not have to write this in italics. I do recommend capitalizing the first letter in the genus, although feedback from other Cambridge graders is that that wasn't necessary, but I would just be thorough with it. I can appreciate not writing in italics because some people naturally write that way, but capitalizing the genus I think is a safe situation. Okay, and that's how the letter, that first one was done for species F. Let's clear this. Moving on to the other ones. It doesn't matter which one you start with now. I'm going to go backwards. I'm going to do E. So we need to identify species E. And again, we're going to start at the top. More than 10 legs are visible, or there's 10 or fewer legs. We got 1, 2, 3, 4, 5, 6, 7, 8. So there's fewer. Go to number 3. A visible tail-like structure or no visible tail-like structure. There definitely is a visible tail-like structure. This is a lobster, and we know that we eat lobster tail. So there is a visible tail-like structure going number 4. Visible claws or no visible claws? Definitely has visible claws. So species E. And again, the genus is capitalized, species is lowercase. This is done and this is done. And same thing, we'll go species D. So get number 1, more than 10 legs or 10 or fewer. Actually that's for eating. 1, 2, 3, 4, 5, 6, 7, 8. That is not a leg. It's not for that. 10 or fewer, we're going to go to number 3. Does it have a visible tail or no visible tail? No tail. We're going to go to 5. Claw of the same size or claws of a different size? Different size. Okay, go to D. I'm sorry, just did it. C. I'm not sure what this one is, honestly. I don't know. For a crustacean. Looks more like a shelled species to me, but I don't know. We're going to start with the dichotomous key. 10 or more or 10 or fewer. I don't see any. No legs. So I'm going to go to number 3. Visible tail-like structure or no visible tail-like structure. This is the tail-like structure. And if you weren't sure, leave this one for last and do process elimination. There's a tail-like structure. We're going to go to number 4. A visible claw or no visible claw. There is no claw. So we are going to put that one here. All right. Finally, B. How many legs we have? 1, 2, 3, 4, 5, 6, 7, 8. These are for feeding. We have 8 legs. So 10 or fewer legs. We go to number 3. Claws of the same, 3 says a visible tail or no visible tail. There's no visible tail here. And so we're going to go to 5. Claws of a different size, claws of the same size. These little claws are the same size. Those 2. Here's B. And then we can identify that A is going to be left over, but just to be sure, it says there's more than 10 legs visible. We need to go to 2. Well, let's count. 1, 2, 3, 4, 5, 6, 7. It's going to be 14 total. There was more than 10, so we go to 2. And then it says antenna shorter than the length of its body. Yes, the antenna are shorter than the length of its body. And there's A. We love a dichotomous key, everybody. Okay, I'll delete that. State 3 features of typical crustaceans. We have to do 3. If you write 4, you will not get any more marks. If anything, you could contradict yourself if you put a 4th here. They will read it. They will read it for the sake of, are you contradicting yourself? So if you said like 2 pairs of antennae, and that's correct. But then down here for number 4, you said 3 pairs of antennae. Well, this is wrong, and it contradicts what you just put up here. So you will now lose this mark, and you won't get that. Because it really just proves like that maybe you don't know what it is. You're, maybe going for like a Hail Mary or something. Just trying to see whatever will work. Likewise, you could have done that for every answer. Erase this more. Okay. If you knew, like, I know there's something about antennae. I'm going to put 2 pairs of antennae, 3 pairs, 4 pairs of antennae. Then you won't get any. All right. I'll give you another example. If you got this correct, this is wrong, and then your number 4 you put in, you got it correct. You wouldn't really get it correct because you get 3 chances, essentially, to say what is typical of an adult crustacean. So you only get 3 chances. So don't just start writing a bunch of things. Be confident in what you're writing. The other one is a carapace. This is from section 4.2. the crustaceans. Carapace, a segmented abdomen. And then the other one is going to be jointed legs. Now these are straight from the syllabus. That's that. It says, when I say straight from the syllabus, it says in the syllabus, identify features of adult crustaceans as, etc. All right, we're moving on to number 3 with our corals. We're looking at the view from the side and then the view from above of three different types of coral reefs. All right. And then we have to label them, identify the types. This is from section 5.2, tropical coral reefs. Okay, and I got blue here just so we can identify really the lagoon, because it is identifying if something is a fringing, a barrier, or an atoll based on its proximity to the lagoon. All right, so I'm just going to identify the lagoon here. No lagoon. Central lagoon. All right. For the sake of this question, if you're wondering, can you also highlight things? Yes, you can highlight things. You cannot highlight your own answer that you want Cambridge to read and grade. Highlighting a picture, a graph, an image that they give you is not going to affect their ability to know what the question is. They wrote the questions. They already know what the question is. You're not altering really anything that's, that they don't already know. That nothing that they're going to be marking. Okay. So I'm going to start with this first one. The coral reefs will start off somewhere like this. This is our fringing reef. And the coral is growing right on the edge, like right on the fringes. I always think of it as like a grass skirt. Grass skirts are kind of fringy. They're going right on the edge of our hard substrate that happens to be imagine a volcano. Obviously a dormant volcano can be active. The coral would not grow there. But they need a hard substrate to grow on. And so they grow on the edge of them where they're still, within a good distance from the water. Like by good, I mean like they're close, like shallow, 20 meters from the surface of the water. They need highlight penetration. So it starts off here, the fringing reef. B is fringing. All right, and then this volcano, or dormant volcano, has like solid basaltic rock inside of it now. It's very heavy from like the magma that didn't come out, or lava that didn't come out, whatever you want to refer to it as. So it starts to sink because it's very heavy, like it's going to sink back into the earth. And as it sinks, our coral do not want to sink very far with it because now they're going to get further away from the surface of the water and they want to be in like a very high lit sunlit area. So our coral are going to actually be growing up as our land mass starts to sink, and that's called subsidence, subsiding, subsidence. And so they start to grow up. And what happens is this lagoon will form in this area here, because now we have this area between our sunken dormant volcano. You can see it went down. And now water's going to fill in there, and there's a lagoon. And that is a barrier reef. This is a barrier reef. The lagoon is a barrier between the island and then the coral. This is the coral. Moana lives on a barrier island. And this coral is going to be helpful. It's going to be like the first line of defense against really strong waves, really tall waves. Finally, our landmass has sunk all the way down. Very heavy, it's very dense. And so our lagoon fills in, and this is called an atoll. And I was taught this early in, like, I don't know, I went to school in Ohio, but like 9th grade, I feel like 10th grade. I remember somebody saying there's nothing in the center atoll. So this is an atoll. There's nothing in the middle atoll. There's no island in the middle atoll. And we still have our coral. Remember, our coral is still growing up. They want to be near the sun, and they'll grow on top of each other. And you can measure coral age and like coral growth rate like we can with rings of a tree. But instead of like looking outwards, we're looking at the rate of vertical growth. And this is called an atoll. All right, the next one. All right, finishing with our coral. They say a label line. Label the mouth, the stomach, and the basal plate. This would be something, if I'm not 1,000% confident, which a lot of percent, we might not have it, we want to use a pencil. If somebody told you, Cambridge won't be able to read a pencil, that's not necessarily true. Like, it's not necessarily true. What we don't want to do is use a pen on something and label here and then be like, oh, wait, no, that's not it. And then you do this nasty, like, oh, forget it, don't do that. And then you know, maybe you correct it. We don't want to do that. Like this is terrible. We're not doing that. We're gonna use a pencil. And if you forget to switch back, oh my gosh, I forgot to switch back from a pencil to a pen, it's not the worst. What I think is the worst is ruining an image that you need to label with a pen and then scribbling it out. Okay, when we do label though, you're going to touch the structure that we're labeling. You're going to use a ruler to make a beautiful line. Tap and hold the compass to measure angles. Cute. Okay, interesting. All right, that's a new feature. Fun. All right. First one is the mouth. Our mouth, I'm just going to highlight it so that you know where I'm going to go, is right here. And so your label line does not have to go perfectly straight. Your label line can be diagonal. It could be vertical up and down. And we're going to label this with a pencil. We touch it with a pencil. And then we write outside of it the structure. However, this label itself, like mouth, it has to be written horizontally. You don't have to put a box around it. You don't need to underline it. We don't want to be doing something like this. Because what is your grader going to have to do? They're going to start turning around your paper. We don't want to be obnoxious. Stomach. I think you know where I'm going with the stomach. Stomach's in here. Stomach has gastrointestinal fluids or gastric fluids or digestive fluids to break down food. And then the basal plate. The basal plate attaches a coral polyp to a hard rocky substrate. Oh, highlight, thank you. Down here is going to be our basal plate. All right. Okay, and I need to do a better job giving an example. I would highlight brackets, highlight brackets, highlight brackets, highlight brackets, because most students are not going to miss things that have lines with them, right? Like describe function of the nematocyst, that has a line. 3 causes of reef erosion, those have lines. But this one does not have a line with it. And it is very common. The most missed exam questions don't have lines with them. They have brackets, and students will just miss them. Just look somewhere else. For the sake of practicing syllabus content, I'm going to go beyond what this question asked about the labeling, and I am going to identify other places and briefly say what their function is. If you already know it, you are welcome to just skip ahead of this. So the outer structure of this, we have the basal plate. And then here we have our like stony cup. When I draw it, I don't usually draw these little things down here that connect. I usually just start it off like this. It is a cup-like structure. It's a stony cup. And by stony, it's made of calcium carbonate. Made of calcium carbonate. That's what it's made of, but what is it called? It's called a calyx. Keep your CAs together. CaCO3, calcium carbonate, CaLYX, calyx. Is your stony cup, and it's what the coral polyp sits in. And then, if I zoom in, all around we have this other layer. Really thin layer right here. And this is the walls of the polyp, the walls of the calyx. And these are called theca or theca, doesn't matter. And those are the thin walls of the calyx. And again, all of this connects to the basal plate. At the top, all of these are all tentacles. Tentacles. I need to make this a little bit thinner. Okay. Tentacles. They have nematocyst on them, like on their first layer of skin tissue or tissue, theca tissue. That is what they have, nematocyst. These are stinging structures. So if you've gone across any exam questions, maybe from the May-June 2025 series, and there was some that talked about the structure of a coral polyp or how the coral polyp gets its nutrition, we had to talk about these. Stinging structures, they contain toxins that can kill or stun or sting. And this is used for 2 things. It's used for 1, predator protection, pred protection. So it's like a weapon, right? But it's also used for feeding, so to capture prey. Okay, and so these literally have like a layer of cells on the outside. Remember, coral's an animal first. I mean, it's an animal. Second too. it's an animal. Has to eat something else for energy. And so these are like their little nematocysts coming out and stinging. Having nematocysts puts them in phylum Cnidaria. The other phylum you need to know is phylum Chordata. But phylum Cnidaria is corals, anemones, and jellies, things you don't want to touch. All right, I'm going to come back to the tentacle in just a moment, but when our coral captures prey, it will take it from the tentacles. Tentacles are then going to put it in the mouth, and then the mouth is like literally just there to receive food. It's also where waste will exit. And then in the stomach is where we have our digestive enzymes, or you can say gastric enzymes, digestive fluids to break down our prey. Well, we know that corals have a unique relationship with zooxanthellae. And so we do have one other feature here. Ooh. So right beyond their nematocyst, they have a layer of mutualistic, means they're going to both benefit from this, producers, an algae, and it will do photosynthesis. It's called zooxanthellae. Zooxanthellae. And so its job is to do photosynthesis. It lives inside the coral tissues or inside their tentacles. They do photosynthesis. So they're going to produce glucose. And our coral is going to benefit because when there's not enough things to filter feed throughout the water and feed with their tentacles, then they are able to still get energy by using the glucose that the zooxanthellae inside the coral have synthesized. Zooxanthellae gets protection. Zooxanthellae also gets access to carbon dioxide. Again, we know that the coral is made from CaCO3, so we do have some carbon compounds that the coral is going to have providing for the zooxanthellae. Again, zooxanthellae does get protected, and zooxanthellae does give glucose to the coral. All right, function of our nematocyst. It just so thin contains stinging cells, or they are stinging cells. I don't want to say it contains. The nematocytes contain nematocyst, but these are the stinging cells. And their function to capture prey, or we can talk about avoiding predation. Three causes of reef erosion. This was also an extended response question. It's a cause and effect of reef erosion. That extended response was from May-June 2025, paper 11. Erosion is any time the growth is less than the loss. Excuse me. Or like we're losing more calcium carbonate than they're gaining. And again, this is from section 5.2. This is towards the end of that section. All right, there is a handful, but they're only going to read the first three. The same rules for list applies. We could do a change in pH. Notice just for shorthand, for the sake of practice, I will not do this on an actual exam though. I use delta, Greek letter delta. That means change in. So when I'm putting change, that's why I'm using the delta. Change in temperature. Could go either way. If it gets too warm, zooxanthellae will expel themselves. If it gets too cold, zooxanthellae will expel themselves. We could talk about predation. also referred to as bioerosion. This is by the parrotfish will do this. They eat calcium carbonate from the corals. Or the crown of thorns sea star is also a major predator of corals. Physical damage. And I'm going to say like what causes this. So this could be physical damage by boats. It's like boat anchors or humans. Separate from like a physical damage that's human caused. Could also do hurricanes or strong storms breaking. Disease. For any living thing and its population going down or its growth going down, you can always say disease, truly. Unless it says don't say anything biotic, but you could always say disease. If you're going to talk about pollution, pollution might be too vague. In fact, the mark scheme says to give a named pollution. So we could say sewage, runoff. Not just sewage, that'd be weird, but like how is it getting there? Sewage runoff. Could say fertilizer runoff. Those cause algal blooms. Algal blooms. And another one is a couple things with sediment. I'm going to say sediment abrasion, like so like physical like sandpaper rubbing up against it. Sediment can also settle in the coral polyp's mouth and block it. So sediment blocking the polyp mouth. And one more. Sediment increases turbidity. That's a Y. Turbidity. Which is going to cause a decrease in light penetration. Light pen. Which is going to cause a decrease in photosynthesis. Which is going to cause a lack of growth. Of zooxanthellae. Or you could say a lack of productivity rather for zooxanthellae. Therefore, which, you know, is going to affect the coral because it does get that energy from the zooxanthellae. The coral does. A lot of them. Yeah, I'm in the front. All right. Our next one. So Unit 4 is section 3.2, feeding relationships. We see a pyramid of energy for marine food chain. So it shows the relative amount of energy in each trophic level. The energy in each trophic level is in arbitrary units, and a pyramid of energy is not to scale, hooray. So identify a tertiary consumer. And quick way to get this wrong is if you read tertiary and you're like, okay, enough, I'm going to go to number 3. I'm just going to highlight my brackets. You say, I'm just going to go to 3, and you go 1, 2, 3. We say small fish. You're wrong. This 1, 2, 3, you're labeling trophic levels. So this is trophic level 1, trophic level 2, trophic level 3, trophic level 4. They want the tertiary consumer. They want the third eater. Here we have producer. First eater. second eater, third eater. 1 second, I'm gonna yell. Hey, Olive. Yeah. Jeremy. Ollie, you have the burger? Yeah, but I don't want that. Okay. D-ham? I guess I'm gonna ask if I can have a sandwich. Okay, it is new. And there's yogurts too. My eye hurts. It still hurts. I'm sorry, y'all. I'll help you in a minute. Okay, sorry. I don't edit these. And if I pause them, I have to like restart this, and I can't stand doing that. Identify the tertiary consumer, third eater, and we are going to put the sea anemone. Anemone. So let's don't get tertiary consumer confused with third trophic level. Calculate the percent decrease in energy between trophic level 2 and 3, and it's 2 marks. Always show your work even if they don't ask. So trophic level 2 and 3, that's the 98. Zooplankton had 98. And then arbitrary units, and then trophic level 3 is our small fish. So in the syllabus, you'll have to calculate trophic level transfer efficiency, and that's how efficient the energy transfer is from one trophic level to the next, or like what portion goes to the next trophic level. Typically, you guys know that there's about 10% that goes to the next trophic level, but you need to be a little bit more precise when you're calculating it for this class. This question isn't asking how much goes to the next trophic level. This question is asking how much was lost, how much didn't go, what was the percent loss or the percent decrease between those trophic levels. All right, and if we do 98 minus 15, and then divide it by 98. Okay. All right, if you divide it, just make sure there's no decimals or anything. You should get 85%. 83 times 98. I said times. Get out of here. Divided by. Okay. You get 0.8469. Okay. Multiply by 100. And then you'll see it in your percent. And it's 84.69. It doesn't tell you what to round it to, but if you look at your other values, they're all pretty much with 2 significant figures. Like 98's 2 significant figures, 15, 83, all those are 2 sig figs. So I'm going to round this to 85% lost. If you're thinking, do you have to say lost? You don't have to say lost. However, if it said the percent change, then we would. Because a number could change by going up or going down. This question is implying that there is a decrease. Just make sure I'm not speaking some nonsense. No. You don't need to show that. You don't need to say that it's lost. If it did say change, though, you will need to say that, like, how it changed. It went up or it went down. one of the marks came specifically just from showing the difference. Like, here's the amount that was lost, and then that's the percent. Like, that would be the second one, is doing the percent, like out of the original. All right, how is energy lost between trophic levels? This also won't change, but it's specifically trophic levels 3 and 4. So we have a small fish getting eaten by an anemone. For every feeding relationship, there's going to be energy loss from respiration or heat. There's going to be, for this one, undigestible parts, like the bones. The anemone probably won't be able to digest the bones. That's typically not what we consider to be biomass. Any parts uneaten. Not that it's undigestible, but they didn't eat it in one bite. Waste or excretion. Movement. The anemone doesn't move too much, but the anemone does have to move its tentacles, and it does retract in and out of its base. Okay, and then finally, this is going to be nutrients. This is section 3.3. On the syllabus, it does say identify, excuse me. Oh my gosh, biological uses for our essential elements: nitrogen, magnesium, phosphorus. Another one is calcium. And then, of course, carbon. All right. First one, so we need to put a tick mark in there. We know for sure chlorophyll needs magnesium. The other one, though, is nitrogen. And I also want to say it's typically in the past, this question has only just been like chlorophyll, magnesium for chlorophyll. That's it. And that's the way it's been, honestly, since I started teaching this in 2017. This is the first year that I've seen them ask for more. And on October-November paper 11, 2025, they had a question, state 2 elements that are found in chlorophyll. And I don't remember the chlorophyll formula off the top of my head. It's something big. But for chlorophyll, you could say, obviously, magnesium, nitrogen, carbon, hydrogen, or oxygen. Nitrogen is a great guess. Nitrogen's in a lot of biological compounds. Protein. Cambridge just wants you to associate it with, there we go, John. And then DNA. We have our sugar phosphate backbone, and then a nitrogenous base. We want the we want the nitrogen and the phosphorus for the DNA. Calcium, CaCO3. That's for our mollusk shells or the calcium carbonate, like the coral skeleton. We could also do calcium phosphate. Calcium phosphate is in bones and teeth. And then carbon is an all organic compounds. Always just glucose on that one. All right. All right, we're close. So after this, well, we have 5, 6, and then we're on to the big ones. Rocky shores are an example of a habitat found in a littoral zone, and it has high levels of biodiversity. 1 mark for our definition. 3 marks for our 3 levels. This is actually a pretty easy freaking test. Notice all these like state things you just got, just almost everywhere. State, like if you don't know it, you don't know it. Like this was a lovely little ditty by Jack and Diane. Littoral zone. This definition is very specific. Same as salinity. Salinity, you have to put concentration of dissolved solids, but you have to have concentration. Littoral zone is very specific. We need to be specific about the spring tides. So littoral zone is littorally the region. It's like it's the intertidal region. There's my terrible shoreline. Girl. Okay. Let's call this my high tide, like my high spring tide. And then down here will be the low tide, the low spring tide. And so the littoral zone is like your space between. It's the area that's going to be most influenced of this, like it's the intertidal area. But when we consider this, we consider like our highest high and our lowest low possible. And that's why we have to emphasize the spring tides. Intertidal region. There's BTWN, between. The high spring tide, height, and low spring tide, height. And if you're asking, do I have to say height? You could say mark. The high spring tide mark, low spring tide mark, because that's what we're measuring about them. We're literally measuring the water height. Littoral zone is from section 2.2. Your other littoral zones, you have rocky shore, sandy shore, muddy shore, estuary, delta. That's it. So those are areas influenced by tidal changes. State the 3 levels of biodiversity. This is section of 4.3. So there's biodiversity in general measures species richness. And that's the amount of different species you have in an area, and species abundance. And that's the amount of individuals in those species. So an example is like a zoo. A zoo has a lot of different species. They have species richness. But a zoo is not abundant. They do not have a lot of each species in like that population. So there isn't a lot of lions. There might be, 4, 3 elephants, when really like in nature, a herd would be much bigger. So that is in general like what biodiversity measures, but there's 3 levels that we can look at it, 3 ways to consider it. And they are genetic diversity. They do not have to go in this order. Ecological diversity. So how diverse is an ecosystem? That could either be local or global. Locally, where I live in Brevard County, we do have very diverse ecosystems. So we have an estuary, or like a mangrove forest. We also have a sandy shore. We have, I can't think of what it goes like sandy brush areas. I don't know. We have a lot. Rivers, freshwater rivers. And then species diversity. Species diversity are the different species, and their abundance in an area. All right. Marine organisms in terms of climate control. So we've talked about climate control with a lot of our producers. Phytoplankton, macroalgae or kelp, seagrasses, their climate control. Climate control has a C, another C. I don't care about both those C's, maybe. Has a C, and the other one is O. I'll try it again. There we go. C, O, and O. CO2. Climate control is all about taking in CO2 for photosynthesis, but ultimately it's not allowing CO2 then to be in the atmosphere, becoming a greenhouse gas or dissolving into the ocean to form carbonic acid and contribute to ocean acidification. So the beneficial role, so we need to frame this as being beneficial, of marine organisms. When they say organisms, it's not necessarily just producers. So we can consider this with plants and with some animals in terms of climate control. So are there any animals maybe that take in CO2 or do something with carbonate ions or carbon ions? The first thing, though, is definitely to think about the producers. Take in CO2, and none of these have to go in this order. Okay, and then the next thing I'm going to say is they're considered a carbon sink, right? Anywhere that we're going to be absorbing or taking in a lot of carbon dioxide, those are carbon sinks. Okay. Carbon compounds. We talk about excreted, like from animal waste. Again, we're going to frame this like instead of it becoming carbonic acid or contributing to climate change, being a prevalent greenhouse gas. Instead, these carbon compounds are now in the biotic system getting excreted as waste or as like marine snow. Or carbon ions get stored in calcium carbonate. Corals or mollusks. So there's less CO2 contributing to climate change. You could say greenhouse gases. Don't put GHGs on your test. Less CO2. Creating carbonic acid. H2CO3. Also section 3.3 with nutrient cycles, and towards the very end, I think maybe 3.3.12, the carbon cycle. Yay, play tectonics. Identify the type of plate boundary labeled A and the plate boundary labeled B in figure 6.1. Here's A. Our arrows moving towards each other, and this is a convergent boundary. And B, they are moving in anti-parallel directions, or sliding past each other laterally. And that's a transform boundary. A is a convergent boundary. B is transform. Now we have state one feature that forms on the ocean floor, a plate boundary labeled C. C is moving apart from each other. And so here we're going to have magma coming up and solidifying on both sides, causing a mid-ocean ridge. But also, there's going to be areas here where the crust is really thin, and there's cracks in the crust, and cold water will seep through those cracks and get superheated by the magma chamber and create hydrothermal vents. But here labeled C, we're going to say a mid-ocean ridge. I'm going to put MOR. Can you write that on your test? Probably. But if you are asking yourself, can I write this? Then write it out then. It's just like, should I be wearing this? Should I wear this to school today? You think this is inappropriate? The answer is always don't wear that to school then. Also volcano. It's too many C's. Volcano happens with any of the features that have the V in it. Not features, sorry, boundaries. Convergent, divergent, because magma's coming out and has to solidify. Tsunamis. After tsunamis, then we are into our extended response. All right, we're going to want to talk about the boundary. It is explained, so we're going to give reasons. It already says how, but why. Give reasons for this. A tsunami at the plate labeled A. And A is a convergent boundary, so this is not a transform boundary. We want to say things like, okay, we have the convergence. This plate right here is going underneath. We're going to talk about subduction. Why? We need to talk about density. And then once we're down here, there's the mantle, not mantello, mantle. Our sinking oceanic crust is going to remelt. Oh, and I'm going too far. I don't need to talk about remelting at all because I'm not going to form a volcano. So that doesn't matter. If I was going to ask about a volcano, I would go into that. We need to talk about pressure building up. Pressure, some energy, like potential energy. It's increasing. And then we need one plate suddenly slips. We need to talk about a lot of energy release suddenly. And once the subducting plate finally slips down, we're going to have, like, literally our seabed like uplifts. Just for a moment, it lifts up to allow this plate to slip down. And when it does that, it also lifts up all the ocean above it. And we're going to say for that, this displaces a large, not amount, it's water, volume of water. All right. Give you a moment. The oceanic plate subducts below a continental plate. I'm going to put PL8 now. Yes, okay, the Pacific plate is an oceanic plate. It's not in the mark scheme. However, the mark scheme does say alternative valid point, AVP. Just from my experience, and tsunamis have been on the syllabus since I started teaching this in 2017, we always want to talk about density. So, like, the explain is why, it's how, give reasons. If I said why, oceanic plate is more dense. Oh, I know, I forgot a word down here. Pressure potential energy increase because they're stuck. The plates, like, they can't slide past each other. They're, miles and miles thick. The plates get stuck or locked. And pressure, or you could talk about potential energy. I think of this like an arm wrestle. There's so much energy in there before, like, the arm wrestling's actually over, but there's so much energy in there you don't necessarily see it. It's just stored energy. Pressure, potential energy increases, or you could say it builds up. One plate suddenly, this happens suddenly, slips. And we don't know which one. Which one gives up? If it's the oceanic, if it's the continental, definitely needs suddenly. One plate suddenly slips. Oh, another one. Causing an underwater EQ, which is an earthquake. So all of these right here is the earthquake, the underwater earthquake. But to get the tsunami, you need to do the earthquake first. A large amount of energy. I'm going to say suddenly again to be safe. Is suddenly released. I'm going to erase some of this. Which displaces a large volume of water caused by the seabed lifting, or you could say uplift of the seabed will displace a large volume of water. It does not have to go necessarily in this exact order, this pretty much this order, but not this exact detailed order. All right. Explain the factors that can change the tidal range in one location. All right, we are in our extended response questions. Yay. Advice, start in this section. So you get your test. If you're listening to this in 2026, you're going to get your test on Monday. No, sorry, today's Monday. Thursday, the 23rd. And you're going to flip it to the back. And you're going to check to see if you have a question right here. And answer. And make sure you just acknowledge it. Sometimes it's on the back page. And we're going to invest our brain in our extended response questions first before we do those short 1, 2, 3 mark questions. And if you feel dumb about a question, leave it. And maybe as you go throughout the rest of the test, you can be reminded of some answers. Okay. This one you can kind of go either way with it. Explain, so why, how, the factors that will change the tidal range in one location. So any location of your choice. Tidal range is a major definition. I know we're going to identify that and we're going to say, girl, let's give a definition. And change. Well, the tidal range is a number. Numbers could go up, they could go down. And so we are going to talk about that. There's not a right or a wrong answer. If you want to frame a factor and like this is something that's going to cause the tidal range to seem higher than it normally will. Okay. I'm thinking about the acronym that I want to say on the internet. So I'll give you the friendly one. Factors that affect tidal height. When you have a whole list of these factors on your syllabus, it's best to make some kind of acronym or mnemonic because otherwise, how are we going to remember them all? And you won't remember them all of everything. Excuse me, there's so much content, it's hard. Okay. So one of the acronyms that we had in class was sharp slopes are so dangerous. And then I looked at this and I was like, oh no, that's donkey D. If you know what I mean. I think my 5th period class this year did Shapely, Sally, Sloppy, Shapely, Sally, 8, 6. Oh no, I don't recall. Strawberries at dawn, or Sloppy Sally, 8, 6 strawberries at dawn, or donkey D. This is for our tides. Factors that affect tidal range. What do they stand for? Okay, this one is going to be alignment of the Earth, Moon, and Sun. So if it's at 90 degrees or 180, is it a spring or a neap tide? Shape of shoreline. Sharp slopes. Slope of shoreline. And this first S, the shape of the shoreline, can also be the coastline. Sharp slopes are, here's air pressure. So dangerous. Okay. This is one of these S's is the size of the body of water, like being influenced by the tides. Is it like a sea or is it an ocean? And then this is going to be the speed of the wind. And this is direction of the wind. All right, and we can frame this however we want. Explain the factors that change the tidal range. We're going to give a definition. All right, so range, it's the difference, and we're measuring the tidal height. So it's the difference in high tide height from low tide height. And specifically, they're not asking for this, you don't have to say it, but I do encourage my students to say it so they can at least get it right if they have to calculate this. It's in 1 day. Difference in high tide height I've seen this where height was underlined. So like a year where height was underlined, and that means you had to include it. Okay. I'm going to start with this first one. So the alignment of the Earth, Moon, and Sun. Bullets for me mean that this is a new mark. Section B should be in continuous prose. That means in like continuous sentence structure. You can give yourself bullet points to keep yourself on track about talking about like different ideas, making sure maybe you get to these 8 marks. You cannot just do like this down here. You cannot just do these random lists without any explanation or linking ideas. They do need to be linked together. All right, I can say what tides are caused by. And if you're not sure, like, can I say this? Well, try it. Try it. You know, you're not limited to what you can write here. They didn't say only 8. You'll write your heart out. This is where you're going to get a lot of your points. Tides are caused by the gravitational, I'm going to put grav, pull of moon and sun on Earth. So this is Earth and Earth's water getting pulled on from the moon and the sun. Okay, and my first one then, so here I got definitions, just normal tide, and then we got tidal range right here. I'm in Earth, Moon, and Sun. And I'm just going to talk about the factors. So when the EMS, Earth, Moon, and Sun, are in alignment, now I don't have to say this one because 180 degrees of each other. I'll finish what I was saying. I'm just by nature going to start off with something that's going to make the tidal range really large. This question does not say you have to talk about making the tidal range its highest. It just says a change. So I could also say the reverse argument. I'm just gravitating towards things that are going to make the tidal range large. Second mark, the tidal range is greatest or largest, right? That's because our high tides are higher than normal. Our low tides are lower than normal. And so the range is going to be greater. And if I said like, well, why? Back to our command word, why and how. Why is the tidal range the greatest? This is due to the gravitational forces or the gravitational pull from your moon and sun being in the same relative direction. They're additive to each other. They're working like together. They're pulling in the same relative direction. Gravitational pull of the moon and sun. I'm going to put M and S. Do not do that on Thursday. Being additive. Like they're working together. All right. This test also did allow you to talk about neap tides. And I also did not call it this first one. When the Earth, Moon, and Sun are in alignment, 180 degrees of each other, times greatest. A spring tide is when, blah, 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 blah. And I could also take this on the side and label here spring, and that would also give me a mark. Truly. Yes, Olive. Neap tides are caused when the Earth, Moon, and Sun are at right angles of each other, or 90 degrees of each other. You know, it's not on, they did put it on the mark scheme. They did put alternative valid point. Often they do, like when we talk about the moon phases. This is during a quarter or a crescent moon. Not boon, moon. In parentheses because it's not directly in the mark scheme. Likewise, I'm going to throw this over here because you don't know. You don't know what the mark scheme is going to say, you know, for this year. This is during a new or a full moon, and you don't have to remember both of them. For example, in class, I teach my students for NEAP. Can write it like this. And there's our little crescent moon. Or, in one word, we have 1, 2, 3, 4 letters. So in one word, we have 4 letters. Quarter moons. Quarter moons give us a neap tide. Okay, almost on the neap tide. Caused when the Earth, Moon, and Sun are at 90-degree angles of each other, or right angles. During quarter or crescent moon. The tidal range, so the difference between your high and low tide, is smallest due to the Earth, Moon, and Sun. Get rid of Earth. The Earth is not causing its own tides. Due to the Moon and Sun pulling, or you could say exerting, a gravitational force in opposite directions. All right, I'm surprised they let you talk about both of those. But I mean, again, you won't know, and there's nothing wrong with writing more than what you think is going to be there, not on these kind of questions. We can just now talk about like all the rest, like things that can increase it or decrease it, however you want to frame it, factors that can change it, increase it or decrease it. So we have like the shape of the shoreline. Our shorelines could be wide or narrow. Okay, and I'm going to say, if the coastline is, it doesn't matter, I'm going to say narrow, like the Bay of Fundy. The tidal range, I'm going to put TR, is higher, like the Bay of Fundy. If you don't know what I'm talking about, Bay of Fundy in Nova Scotia, Canada. You can look at Bay of Fundy GIF, or like just a short video. It has the highest tidal range in the world. So during low tide, like they cannot be, like it's dry. Like you cannot take your boat back into dock during low tide. You have to wait during high tide. But I could also say if the coastline was wide, the tidal range will be lower. But this one is where it's a true or the reverse argument. You can't get 2 marks for both opposing things. Okay, we can talk about the slope. If the slope of the coast or shore is steep, the tidal range, TR, is high. Or the reverse argument is that if the slope of the coast is gradual or small, don't say flat, because that's not reality for the coast, but gradual or small, the tidal range will be lower. That's like how it is on the east coast of Florida. All right. And then we got air pressure. Or atmospheric pressure, same thing. Atmospheric pressure is high, right? So we're going to have a lot of air pushing down. It's going to keep our water down. Although like during a hurricane, if we had a release of that air pressure, our water is able to like expand a bit and it'll make our tidal range look higher. But if the air pressure is higher, the tidal range is lower, or the reverse argument. And again, that's because pressure from the air, think like sitting on top of a suitcase and you're trying to smash the clothes in the suitcase and smash the suitcase closed. The suitcase is like the ocean and you are the air pressure just trying to smash it down. If you take that pressure off, it can expand. Size of the body of water. If the size of the hold on, I just have a thought. Okay, like an ocean, if it's large, like an ocean. The tidal range is higher because you're moving in more water, or the reverse argument. Okay, and then we got 2 things about wind. So it's the direction. I'm going to talk about the direction first, and I'm going to talk about the speed. Because the speed is kind of like irrelevant until you talk about what direction it's going. If the wind is blowing onshore, you don't have to say onshore, you could say offshore. But if it's onshore, TR, tidal range, is higher because we're blowing water onto the shore. Or the reverse argument, you could say if the wind is blowing offshore, the tidal range is lower. And finally, if the wind, quotes mean whatever I wrote above it, bring it down. Okay, if the wind speed is greater, so it's onshore and it's greater, tidal range is higher. So now we're just blowing even more water onto land. Okay, the reason why I stopped before is because I was thinking, I remember doing this in like January, and there's some marks that I wrote down. It's good for practice. It still supports the factors that affect tidal range, but let's look at the question. In one location. In one location. So this is going to be like a known location. A known location. It can change this in one location. We got to think about things that really aren't going to change. Now, our Earth, Moon, and Sun situation, they change, and they do change. That's fine. These are fine. These are fine. The coastline. The coastline doesn't change. If the coastline is narrow, the tidal range is higher. But that's not something that can change. So this really wouldn't apply. It doesn't change. If the slope of the coast is steep, the slope is pretty much going to stay the same too. Then the tidal range can be affected. But different locations can have different shapes of coastlines and different slopes of shorelines. This doesn't apply because this isn't going to change. It's not like, in Florida we go out and we're like, oh my gosh, all of a sudden this is like a very narrow coastline. No, it's like wide sandy beaches. Air pressure can change. That has to deal with like the climate. Size of the body of water, that's not going to change. It's not like all of a sudden the Atlantic Ocean is not influencing tides on the east coast of Florida. And I'm using Florida as the example because that's where I live. Like in England, it's not like the Atlantic Ocean is not going to be affecting England. Like, oh my gosh, what is this? The Gulf, like the Gulf of Mexico now is on the, like where the Atlantic was. That doesn't happen. So the wind can change and the wind speed could change, like the wind direction, the wind speed could change. But these three that I circled, they're not going to be applicable on the mark scheme because they don't change. They're not changing. They can be different in different areas, but they're not something that can change in one location. Okay, the red mangrove tree. And then we got uses of the red mangrove tree, and then a great one about abiotic factors. Can I just get a dang brown? Okay, the most prevalent thing we think about a mangrove are its prop roots. Okay, and they grow in the water. The substrate is very fine. Small sands and silts. This is an estuary. If you live on the east coast of Florida, maybe you have the Indian River Lagoon, or perhaps you call it the Intracoastal Waterway. Lagoons, estuaries, bays. Those are all going to be the same thing. Small sediment. And it can sometimes smell here. Like sometimes it's like marshy. It's not a marsh. But if you live in a place that there is like a salt marsh, sometimes it can smell. It's very small sediment, has low permeability. So it's hard for things to go through it. When I say things, I mean like gases. And that's what makes things smell bad. Okay, these are our prop roots. They're for stability more than anything, but they also will slow down the water and cause sediment to deposit. Moving up. We got the rest of the plant. Okay, I have it here too. If we look really close, look at those thick leaves. They're so thick, you can't see through them. They're very thick. All of your marine plants are going to be very thick. They also look like they have debris on them or, I don't know, pests. But your marine plants that grow near coastal estuaries or like the ocean, they're going to be very thick. They have to because they have to stop salt from coming in. You also have to stop water loss. You see these right here, those little salt buildups, all those little white sediment deposits. It's like salt. Because salt water will will blow on here, and then it's the salt that's going to stay piled up. It's not going to be permeating in. And we got a seed, And the seed is growing its little baby, its little propagule. You can see it coming out of the seed. You don't see a full-grown one yet. Not on this picture. And that's going to be the next thing that I will include. So we got our little propagule. Our propagule develops from a seed. It gets its nutrients and water from the parent plant, and it develops on the parent plant. This seed would not last. If this seed fell off, it would not last in the salt water. And so this is called viviparous reproduction. The structure is called a propagule. Propagule. And it's produced through viviparous reproduction, which is really like you're growing your offspring on you. Viviparous reproduction, instead of like just dropping down a seed, that seed would not live in the salt and fresh water. So it gives it a chance. It gets nutrients and water from the parent plant and starts to develop on the parent plant. So when this like drops off, it's already got its prop roots ready to go, and it will float for quite some time until it finds a suitable sediment, soft sediment that it can start growing in. But that also helps it develop in new areas and limit competition with the parent plant. Because, I mean, there's not space down here for more prop roots. Okay, we need to do gas exchange. So even plants need to get oxygen, of course, because they have to do cellular respiration. It's really hard to get gases whenever you're in this very fine, very small sediment down here. Gases do not come in and out easily because it's just so, the porosity, the pore space between these sediments, the gaps between the sediments is so small. So we have these little structures on the roots, and they allow the plant, the mangrove, to get gas from the atmosphere. And the gas I'm talking about is oxygen. Gas exchange from ATM. And these are called dang it, hold on. Not denticles. And I was going to say vesicles. No, that's in the cell. It's lenticles. Lenticles. They're like little nostrils. Little nostrils. It's like a witch's nostril. You draw a little pig nose. Or a little dog nose. Little nostrils. So yeah, they can take oxygen in from the atmosphere because it's not coming in from this tiny little sediment. All right, and these roots, when they are like a fence, they will slow down water. So that also helps stop with erosion because it slows down water currents. They're also like a line of defense for waves, but this isn't like an enclosed area. Partially enclosed area, I should say. So there's not big waves here. Again, if you think about the Indian River Lagoon or Intercoastal Waterway, whatever you call it, those areas are enclosed and separated a bit from harsh waves straight from the shoreline. But this will slow down water, and if this is like a snow globe, if water traveling is like a snow globe with all the particulates suspended throughout it, when it slows down, those are able to deposit. Those sediments are able to deposit or do sedimentation and build up. And this is good because this builds up the sediment around the structure itself, around like the prop roots itself. And it does need soft sediment to grow. Okay, so the three things that we need to talk about for the mangrove is going to be its prop roots, being able to do gas exchange, offering stability. Prop roots also are impermeable to the salt. You don't need to say how, you just need to say that they are. They do not allow salt to come through. And we know that they are in estuarine waters, so they are in a mix of salt and fresh water from rivers. And our roots are down here, but no salt gets to go through. We can talk briefly about the leaves and then the propagules. I'm going to split this into 3 things just to keep myself organized. And you can also do the same on your exam. You can split this into 3. Okay, thank you. Good day. 19. Okay. You can also do this on your test. You can split things up with a T-chart or, I don't know, a tri-chart. For the roots. Now what I'm doing here, you won't do on your test because we cannot highlight the words we're going to write at all. I'll do the first ones first, the leaves. Any marine plant is going to have leaves that are tough, thick, or leathery, and it's to prevent salt from coming in. Salt intrusion, or you could say salts from entering, or to prevent water loss. Just literally from being around like a saline area and like wind. And if we think again about our command word, it says explain. So we need to be explaining. So this is why I put the Y. That's it for the leaves. Prop roots. You will get a mark just for mentioning prop roots, like just for saying them. So notice I'm separating them by bullet points into the marks that they're given so you can see like where the marks are at. But on your exam, you will want to write this, maybe like with more continuous prose where these marks are going to like lead towards each other and they're not just short, I don't know, I say it's like caveman talk, like prop root, stability. We need to like link it together. This substrate is easily erodible, so having the stability is helpful. The roots also slow down water to allow sedimentation or to allow an increased deposition, like the depositing or sedimentation of sediments. Okay, and that's good for the stability portion. Let me lock a screen. Where? I never do this. Okay, great. Stability. Gas exchange. Okay. They do not have to go in this order either. The substrate has low oxygen. Just for your own sake, why? Because it has low permeability of gases. This isn't on the syllabus. I'm sorry, this isn't on the mark scheme. It's on the syllabus. It's not on the mark scheme. It could be, but just for the sake of you to know, like, why I'm writing this. Due to small sediment sizes. You could also refer to the water being hypoxic, having low oxygen. Lenticels, which they had in parentheses, which means you did not have to say it. Does that mean you never have to say it? I don't think so. But in October, November, you did not have to say specifically lenticels, but lenticels on the roots. Absorb O2 from the atmosphere. And as I said before, if we're talking about oxygen, definitely talk about respiration. To do respiration. Okay, these things here are all the adaptations for gas exchange in a low permeability substrate. Gas exchange. And these are still structures that are within the roots. All right, and then we can talk about the salt exclusion. The water they live in, the water is estuarine. Sometimes you want to say brackish, and I'll always say, what does that mean? We'll want to say it's a mix of salt and fresh. The roots are impermeable, which means salt cannot permeate through them. The roots are impermeable to salt, or salt is impermeable, to salt, yes. And they're able to so salt is not going to be able to intrude upon the plant and its tissues. Most plants cannot do this. They cannot tolerate growing in salt water. Because of osmosis, they will lose water, but their root structures are impermeable to salt. Does not let the salt come through. They're able to filter in fresh water. Filter through fresh water. All right, and that's the last one. Salt exclusion. Finally, we have our propagules. Or you can head the section with viviparous reproduction. Propagules provide, you could say perform, viviparous reproduction by creating propagules. I could describe them as like seeds that germinate on the trees, like they start growing on the trees. Propagules get nutrients and water from our parent plant. Propagules can float They grow new areas, in new areas, or they can colonize in new areas. Away from the parent plant, which limits competition. Intra-specific competition. I'm going to include that. It'd be intra-specific because it's the same species. They would be competing for space, also competing for nutrients, light availability. All right, and then the roots will grow when they hit a suitable substrate. I have a lot. This is the last section, 5.5. I have a lot on this topic of mangroves. One second. Hey, Ollie. Olive. Yeah? How's your puzzle? Good. I took a break from it, though. Okay, I bet you're doing great. I know. All right. Explain the importance of mangrove forest to humans and to the environment. So this kind of opens it up. 7 marks. We can talk about humans. We can talk about the environment. Okay. Couple of things. Instead of just having to think about, what's important about mangroves? We're going to think about a couple of things. From section 4.3, I keep having my students go back to why do we care about marine biodiversity? What's the importance of maintaining it? Why do we care about conserving species? That whole idea. Because then we can link that to a lot of other ecosystems and why we care about them, what kind of services we get from them. And then the other thing we're going to think about is trees in general. Okay, so from section 4.3 are... Sorry, I need to stop yawning. Okay. I don't have a good acronym for this one. I don't. But for marine biodiversity, I just remember it's MIC P3. Right, there's 3 Ps. And so these are the reasons why we care about biodiversity. The M is going to be it maintains stable ecosystems. So not necessarily applicable to just one mangrove species, but in general, we care because we want to have environments with a lot of different types of species, because that would make it very biodiverse. And if one of those species gets a disease or dies out or can't tolerate a change in the environment, if it's a biodiverse area, we will maintain a stable ecosystem. Our ecosystems are not going to fall apart. Now, is that directly applicable to mangroves? Not directly applicable, because we're really only talking about one species. I continue. C is our climate control. If it's a plant, and it is a mangrove, we're going to talk about climate control. Here's COO. We're going to talk about CO2. That is a service. It certainly is a service, because exasperated storms or stronger storms due to climate change is going to cause a lot of damage, and it's costly. Changing in just your major seasons and temperature ranges could be costly, especially if you're growing crops or you're expecting certain types of animals to be present and they're not. And then we have 1, 2, 3. So 3 things start with letter P. We are going to provide, and protect. We are going to provide medicine. An example from the syllabus will be the keyhole limpet hemocyanin, and that's used for a lot of vaccines, autoimmune therapy. I know there's more. Marine cone snail, I believe, is the wound healing one. The marine worm is schizophrenia medicine. We want to have a go-to medicine example. Providing food. Now, this is food for humans, but this is also like providing food for our food. Like, we want small fish to be able to feed our grouper because we want to eat our grouper. Okay, and then it protects shorelines. Having biodiverse places like healthy corals, mangroves, kelp forest help decrease wave height and slow down water, which decreases shoreline erosion, and that is a $1 billion problem for us every single year. Trees in general. We use it for timber, biofuel, to literally like burn, be a very expensive thing to burn, and in Florida, like illegal. But there we are. Something to also consider is like ecotourism. We're going to benefit from that. That's like the human community thing. Ecotourism. If we have healthy environments, we're going to have animals there. People will want to come here, spend their money to come see all those. Specifically, mangroves are going to help like with their own. It's not the same as protecting shorelines, but when we talk about they're going to slow down water, that's also going to allow deposition of sediments. So the settling of sediments, and that's going to make the water clearer, less turbid, and so we'll be able to have more photosynthesis occurring, which our food sources need. All right. Do this one real quick, and then we'll be with our last one. All right, in general, we can say mangroves. We know mangroves are nursery grounds. And I have a lot of lectures on just mangroves. Ollie, you good? Olive? Yeah. Are you good? Yeah. Okay. For juvenile species, like our future food. This allows biodiversity, or like our organism populations to be stable or maintained. It allows for the growth of human food sources. So when we talk about juvenile fish, we're talking about our future food. Allows for growth of our food sources and employment or income for fishing industries. Again, these answers do not have to go in this order. I'm going to talk about their prop roots, allowing for good conditions for our producers. Prop roots slow down water, or they trap sediment. Which prevents turbid conditions or turbidity. for corals. Remember, corals like clear water, but they also don't want any sediment to settle in their little coral polyp mouth. Seagrasses also want some clear conditions, and they cannot deal with fast-moving water. I mean, they can deal with it, but not constantly. They will get eroded. Decrease in turbidity equals an increase in photosynthesis. And I'm going to say biomass production. Biomass is like what, like the food that we will eat. So like not the shells of like sunflower seeds, but like the seed itself. Biomass is like it's bio, so life, mass, stuff. This is the stuff that is actually like food sources for us. So not bones, not teeth, not fur, not feathers. Those things are not biomass. Okay. Mangrove forests allow for an increase in ecotourism. And money for coastal communities. Often people ask, can I use the money sign? I wouldn't. This is an international exam, and that's the money sign for the dollar. And not everybody uses the dollar. I know England is still using the pound. So don't do that. They're used for timber or fuel. They put them in the same mark. You don't know if that's going to happen. That's why we write more than just 7 things. Can be sources of medicine. In section 4.3, again, biodiversity, there's a whole list of examples of types of medicine and pharmaceuticals we get from marine species. There isn't one specifically for mangroves, but I mean, if we're going to say like marine cone snail schizophrenia medicine, I'm sure there's some from mangroves. You just don't have to be as specific with that on this question because we don't know any from mangroves. Unless you do. Medicine or even like science research, they say. They act as carbon sinks. Or you can say they help provide climate control. By absorbing CO2 for photosynthesis or during photosynthesis. They're a great source of oxygen. They provide oxygen for respiration. They provide coastal protection, or they protect our shorelines by decreasing wave height and water speed. Okay, this is kind of like a cluster one. There's a lot we can write about this. In class today, and this is 2026. We kind of did a big brain map of this, but it's explaining the abiotic factors. I really think this should say processes. I think it should say processes. I digress. That affect the chemical composition or the chemical makeup of seawater. If we're affecting it, we can either increase the chemical composition or we can decrease the chemical compositions that's in there. And then they say, give named examples with reference. Refer to named chemicals. It's any chemical, right? Like water is a chemical. I'm going to scroll down here and I'm just going to sketch some things out. So if we got to simplify this. This is very similar to 3.3 nutrient cycles. And when we considered factors or things that are going to bring nutrients in or make them available, it's a lot of the same kind of answers. Okay, but if we just kind of think about it, there's only a handful of things that really are going to affect the ocean. Volcano. Some runoff. There's runoff. Volcano. What's at the bottom? And we can have some HTVs, hydrothermal vents. The dreaded low power mode. I'm going to be done with it. All the motivation. Okay, yeah, so we have like things that are up here. Like these things can come in, right? It's not just like these things. It could also be like these things. We have things that are on the bottom of the ocean and then things that are on the sides of it. We cannot talk about biotic things. So we cannot, like things that are going to add chemicals in that are biotic. We are not going to talk about decomposition, like the breaking down of organisms, and then we're going to have those chemicals be into the water. We're not going to talk about respiration because that is a biotic thing. We're not going to talk about photo. Again, it's a biotic thing. We're not going to reference marine snow because that is biotic. Okay, Moss. Any excretion. Now we can talk about ways like CO2 is affected in other ways, but just not that. It would be a great thing to do anyway to like mind map this out and not just start writing some craziness. Okay, we can start up here, the top. So that one would be more like atmospheric dissolution or atmospheric gases, like CO2 or O2. I really don't need that second line. They dissolve into the water. Things that can cause them to dissolve more. We can have wave turbulence. Factors in general that affect gas solubility. This is section 1.2.10. We have weight. That's actually smart. Weight, that's actually smart. We got water pressure, temp, atmospheric pressure or air pressure, and salinity. Now it doesn't matter. Getting back to the question, it says factors that can change. We can talk about chemicals being able to stay dissolved or not stay dissolved, like stay in the ocean or not stay in the ocean. It really doesn't matter. You just got to have it right. Wave turbulence will increase gas dissolution, just more kinetic energy. Water pressure. If we have high water pressure, it equals an increase in gas solubility. This like bottom of the ocean has the highest water pressure, obviously, and this is the same example of like pressing down on a suitcase. Temperature. We drink our carbonated things cold because our gassy things need to be with low kinetic energy. So temperature being low, that will mean we have higher gas solubility. So those chemicals that are gases are able to stay dissolved. Atmospheric pressure. The higher the atmospheric pressure, we're going to have higher gas solubility. And I could have talked about any of these with low gas solubility. It just literally doesn't matter. And then if salinity is low, we're going to have higher gas solubility. And in my head, I just say like there's more space for gases to be dissolved when there is less dissolved solids in the water. We do have other things that happen from the atmosphere, like this. So we can have precipitation. Now that's going to affect salinity. Precipitation, salinity is going to go down. Why? Because we're diluting the water. We're adding fresh water. Other things that will make salinity go down. Runoff of fresh water. We can also have melting of sea ice. Salinity increasing. Runoff of fertilizers. I don't want to say agricultural waste. I don't want to say that because that's now biotic. I'm talking about like cow waste. We're going to say fertilizers. Another one is going to be evaporation. We already have precipitation. And then forming sea ice. Fresh water gets removed. When we're talking about salinity, this is one of the first questions that we did. Those chemicals, we need to give examples. Sodium chloride, calcium carbonate, magnesium sulfate. We have gas examples there. All right, what can cause, like, if we were talking about like the runoff, what can cause that to, I guess, have like a greater effect or dissolve at a greater effect, dissolve at a greater rate? It could be a higher water temp. There's an increase in salt or solid solubility. So in class, I talk about like hot coffee versus iced coffee. Iced coffee are going to have to stir. Iced coffee does not have like its own like high kinetic energy because it's iced. The water molecules are going really slow and they are not having as many particle collisions with the sugar molecules to break them down. This is the same thing as like water and salt particle collisions. If you have hot water, hot coffee, those particle collisions are going to be happening on their own just by nature of the water's own kinetic energy. Okay, so we did talk about our runoff from the land. How about this? We got volcanoes. Volcanic ash. We can also have volcanic gases. Volcanic gases may be like hydrogen sulfide. It also comes out of a hydrothermal vent. Hydrothermal vent has H2S. Hydrogen sulfide. You can also say sulfates, sulfur dioxide, I think hydrochloric acid, HCl. All those things will come out of the volcanoes as well. So the ash will get dissolved. It can also get dissolved in the rain, but the gases will also get dissolved into the ocean. Hydrothermal vents at the bottom of the ocean, same thing. And let's see if there's any more. CO2 also comes out of the tectonic activity. Okay. One more I think we could talk about is like upwelling. Brings up solids, or I guess nutrients, but also solids. This could be the nitrates, phosphates, calciums, magnesiums, carbonates, things like that. And what can increase it? We can have an increase in offshore winds. And, oh, there's another one. So calcium carbonate in shells and in corals. However, if we have a decrease in pH, we can start to erode this, and then we will have an increase in calcium ions and carbonate ions in the water. This is crazy, but this will also get marks at the worst case scenario. Okay, and the last thing we're going to put then, I'll try and make this make sense. Looks like dissolution or dissolving of atmospheric gases, like CO2, oxygen. If you want to say nitrogen, you can also say nitrogen. Is increased with, so like the chemical composition is increased. It's increased with wave turbulence. Low temp increases gas. I'm going to put SOL, solubility. Or reverse argument, that's fine. Low salinity increases gas solubility, of course, reverse argument. High atmospheric or air pressure. And this mark, they also included the high water pressure. Will you know that? No. So on Thursday or whenever your exam is, you are just write it separate. You don't know. Okay. Runoff of fertilizer is not biotic because it's not alive. It has to be taken in by a plant first. Fertilizer will increase the nitrates, PO4s, phosphates, magnesium ions, calcium ions. Okay. Runoff of fresh water decreases salinity. We can give examples. The salts include NaCl, magnesium sulfate, CaCO3, calcium carbonate. More decreasing in salinity. So we have a runoff of fresh water. Got it. Melting of ice. Precipitation. Increase in salinity. We already talked about our fertilizer. Could say Actually, we did not specifically say that. It just the runoff of fertilizer increases the nitrates, blah, 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 increased salinity caused by runoff of dissolved solids like the fertilizer. Sea ice formation. Also evaporation. Factors that can affect this, well, if an increase in temperature causes an increase in solubility of salts, and it's not in the mark scheme, but I don't see why it couldn't be due to high kinetic energy and an increase in particle collisions. And if anything, it's just good practice to remind yourself to write it down. Oh, my hand hurts. Volcanic ash and gas can contain hydrochloric acid, hydrogen sulfide, sulfur dioxide, CO2, magnesium ions, hydrothermal vents can increase, like all these same things. Sulfur dioxide, magnesium, sulfates, phosphates. A decrease in pH can increase the calcium, I think it's Ca2+, but it's fine because it's not chemistry class. Ions, the carbonate, the CO3, carbonate ions. By increasing dissolution of CaCO3 shells. Oh gosh, my hand hurts. Offshore winds can increase chemical upwelling. So essentially we highlight everywhere that we gave the example. It says with reference to named chemicals. So here's one. Here we got our nitrates, our phosphates, the magnesiums, the calcium ions, the salts, hydrochloric acid, sulfur dioxide, hydrogen sulfide, carbon dioxide, magnesium. Same things here. This is a big question. And honestly, I really think it should have said processes other than factors. However, we have to at least attempt it because when the graders will look at this, then it's hard to tell like if anybody was misunderstanding something or was the question written strangely, or maybe it could have been written in a more direct way. A word could have been changed. They can only really identify that though and assess themselves if there's something to be assessed. So leaving blank things sucks. Do not leave things blank. Blank test fail. Blank test fail. Say that. You've done a ton of work this whole year. You do not need to leave anything blank. Put something intelligent, anything intelligent. Do not leave things blank. You owe it to yourself. Blank test fail. You just automatically just give that L. Don't do that. Attempt it, and then if they can at least see like, oh, I wonder why, they would write this. Is anybody else getting these kind of answers? They can at least assess themselves and maybe see if maybe they wrote a little bit of an ambiguous question, and then they will amend a mark scheme a little bit to allow this to fit actually what was being asked directly. Oh, thanks for hanging out if you did, and good luck on Thursday.